Hey everybody, I'm back after an annoying interruption from telemarketer. Anyway, I told him to go away. Just a note for all you future YouTubers out there: clear all the phones out of the house before you start recording. Anyway, I was talking about David Nelson's inaugural lecture to start the investigators meeting. He started talking about the title was Acute Requirements for FMR1 in Mouse. But basically, the theme of his presentation was his experience with these conditional knockouts for Fragile X. So these are mice where you can turn the gene on or off. Not both, by the way. You either have a conditional on or a conditional off, but you can do it either way. And of course, the more interesting experiment is if you have a knockout mouse which develops with the gene turned off, and then you subsequently turn it on when the mouse is, say, oh, I don't know, 4 weeks old. So this is the equivalent of childhood or adolescence for a human, and you want to see if you can get that mouse back to normal by turning the gene back on. And essentially what they found in this research is that yes, you can. If you turn the gene back on, most of the symptoms that they see in the mouse go away. Now you have to issue the usual caveat that mice are not people, and that may not be the same in humans. And of course, none of the treatments that we're talking about are quite as good as actually restoring normal production of the gene. But still, it's an impressive result because I think a lot of people expected that Fragile X would be primarily a developmental gene, and so most of the problems would originate during embryogenesis before birth, and a lot of those problems would not really be treatable later on. And it turns out that's not the case. It seems like a lot of the problems, most of the problems, maybe all the problems, are treatable later on in life. The exact timing is hard to work out in mice because mice aren't people and the development is different, but it's very encouraging news. And certainly when they did a similar experiment in Rett syndrome and turned the gene on much later in life and found the mice got better, well, that was a worldwide sensation. The same thing applies in Fragile X. The difference is we actually have treatments that can fix most of these problems, whereas in Rett syndrome, they're still looking for actual therapeutics. So after David, Mark Baer talked about the pathophysiology of Fragile X, and of course, Mark's claim to fame is the mGluR theory, and he talked about mGluR5 antagonists and rescuing the knockout mouse by crossbreeding it with mice that lack the mGluR5 receptor. Essentially, if you cross the knockout mouse with a mouse that has half as much mGluR5, which is a good genetic proxy for treatment with an mGluR5 antagonist, lifelong treatment, then you find that you can rescue just about all the phenotypes. You can essentially cure all the symptoms of Fragile X by doing that. Nope, there goes the phone again, but I'm just going to ignore it this time. Anyway, we then went on to Ben Ustra talking about the rescue of Fragile X behavioral phenotypes and neural protrusion morphology by mGluR5 antagonists. And Ben's lab has actually done some fairly close collaboration with the folks at Novartis and has also tested the available mGluR5 antagonists like MPEP and Phenibam. And they've found some very interesting results. He's also looked at some phenotypes that are found in the cerebellum, and that's the one area in the brain that you really can't fix with mGluR5 antagonists. The cerebellum doesn't have any mGluR5. It has a lot of mGluR1, but it doesn't have any mGluR5. So we're thinking that whatever the symptoms that result from the effects of Fragile X in the cerebellum, they're probably not going to be addressed by mGluR5 antagonists. But we are working on a number of other treatments that work on the signaling pathways, and these are the same signaling pathways throughout the brain. They're not the same receptors, but what comes after the receptor is the same everywhere in the brain. So things like lithium should work everywhere in the brain. Things like minocycline should work everywhere in the brain. A lot of other treatment strategies that we're looking at, like GSK3 inhibitors and ERK inhibitors, should work in the cerebellum just as well. And ultimately, as we discussed toward the end of the meeting, we're looking at developing cocktails of drugs, because no small molecule can fully compensate for the absence of a large protein, complex protein function, like the Fragile X mental retardation protein, FMRP, that's missing in Fragile X. So we're potentially looking at cocktails of treatments, but certainly the cornerstone of all these combination therapies would be something like an mGlu5 antagonist. And so the evidence that was presented was pretty compelling. So we started off with a bang, and I think that got everybody pretty psyched up. And we then moved on to some of the other folks that are working on specific translational research that's looking at treating various aspects of Fragile X syndrome. Tom Jongens is one of the fly biologists, and he presented some of his previous research as well as some of his newer research talking about rescuing the fly phenotype. And interestingly, fruit flies have cognition, they have learning and memory, which by itself is surprising, and the Fragile X mutant flies show deficits in this learning and memory. And it's all centered around courtship rituals. And it's something that fly biologists are quite good at doing, and you can get a quick, easy, and relatively cheap readout and judge the effect of potential treatments a lot more quickly than you can do it with a knockout mouse. Mice cost a fortune for housing, and they take months, if not years, to breed enough of them. And surprisingly, they don't actually have good cognitive phenotypes. We don't see a lot of difference in learning and memory between Fragile X mice and their wild-type litter mates. But we do see some differences, and we do see some things that are amenable to treatment. And I'll probably talk about some of those things later on. Tom Jongens also showed some very interesting convergences between Fragile X and Alzheimer's disease models in flies. And that was something that would crop up again as we started to talk about some of Kara Westmark's research, where she's found that amyloid precursor protein is regulated by the Fragile X protein, and it's regulated by mGlu5. And so people with Fragile X and mice with Fragile X have too much of this amyloid precursor protein. And if you give them mGlu5 antagonists, that normalizes those levels. Now, people with Fragile X metabolize their APP normally, so they don't tend to have the plaques and tangles that lead to Alzheimer's disease, the abnormal deposits of the metabolic byproducts of APP. But they do have too much APP, and that in and of itself may be significant and may affect function. Paul Lombroso from Yale talked about his work with a regulatory enzyme called STEP. It's a tyrosine phosphatase that is responsible for internalizing AMPA receptors. It internalizes the AMPA receptors in response to the signal coming in through mGlu5. So if you could inhibit this enzyme STEP, you could potentially ameliorate Fragile X by increasing the amount of AMPA displayed on the cell surface, the AMPA receptors. So it would be like administering an ampakine to somebody. This also has a lot of applications in Alzheimer's disease, and it turns out that STEP inhibitors are being actively investigated as drug treatments for Alzheimer's and a number of other problems.